All right. Well, it seems like we have more or less a quorum. Again, welcome to the uh, December version of the Bioinformatics at Home webinar. This is a real treat. It is actually not a Zoom background. So, welcome. And yeah, we'll just go ahead and jump in. Again, any questions, pop them in the chat. Let's go. All right. So, I'm Ben Busby. I'm a Solution Science Director at DNA Nexus. If you need to find me, Twitter is a good way to do so. I run a lot of hackathons too, so if you're interested in that, check them out. I'm just, if you see my cursor moving, it's because I'm moving the chat boxes out of the way so I can see my slides. Anyway, here's a hackathon I ran here in Pittsburgh at Carnegie Mellon about a year ago. By the way, if you're interested in hackathons, we have a Bringing Genomic Data to the Clinic hackathon at CMU, obviously virtually this year, January 6th to 8th. So if you're interested in that, let me know. I also do a lot of bioinformatics education. This is my friend Jason Williams. He's sort of a world expert in bioinformatics education. If you're a bioinformatics educator, please reach out to me. There's a lot of international groups that collaborate on bioinformatics education. I'd love to put you in touch with those. So you can find me on Twitter, direct message me, bbusby at dnanexus.com. I think that's at the bottom of the screen. Yeah. And also, if you have ideas, questions, comments, concerns, you can also email homeseries at dnanexus.com. That's the email alias for this webinar series. Awesome. So, administrative announcements aside, what we're going to try to do today in the next about 40 minutes is really sort of go and talk about population genomics all the way from people who are just beginning their sort of journey and looking into this and thinking about how that works, all the way to some of you are probably advanced theorists. You've been doing GWAS for a long time and really thinking about the new and next. And I'm going to talk about sort of worldwide consortia of people that are finding variants that are tied to phenotypes, how we, some prototype work that I've done in sort of large collaborations with a bunch of researchers, thinking about how to share information based on these variants, and I'm going to try to convince you that that's really important. And I'll also talk about some more sort of traditional population genetics stuff, talking about GWAS, VWAS, and really I'm going to talk a lot about cohort selection. So we'll jump into that. But I think the fundamental problem in population genomics, a lot of the algorithms that we use have been around for 10 or 15 years. But I think the big problem has really been in activation energy. As all of you know, I mean, you have to have sort of, or heretofore, you've had to have sort of, some command line savvy and so on and so forth to run GWAS. But then you also need, you need domain experts, biologists, clinicians to help you make the cohorts. And then you got to think about covariates. So there's a bunch of statistics in there. And that's really resulted in a big learning curve. And so I don't think we've been able to apply population genetics techniques as much as we could. And also, the data hasn't really been there heretofore. And so, then now we're at the advent where we have better data and better tools for population genomics. By the way, I see a bunch of people just walked in the door. If you have questions, comments, concerns, pop them in the chat. And yes, the slides will be available. All right. So I think what really we're on an advent now of reducing that activation energy and really making population genomics, really allowing lots of folks across the bioinformatics and biological and clinical spectrum to be able to contribute to GWAS. And large data sets like UK Biobank are coming online, but also really sort of simpler, easier to use, more approachable analytical techniques. And I'm going to talk a little bit about that as we go through today. And I think that these subtle barrier reductions in bioinformatics are really critical. And I think that obviously the biggest one is probably BLAST, right? I mean, in the 70s and 80s, people used Smith-Waterman alignments. So really what that meant was that really sort of super computational people without access to large compute were locked out of bioinformatics. And when BLAST came online, now everyone is able to do some super sequencing bioinformatics. I know that sounds probably like a fairly trivial example to some of you. Here's a more nuanced one, and I'd say that comes out of evolutionary biology. That's an algorithm called FastTree2, which really exploits fast searching of novel space. And why that's important is because algorithms like FIML and Raximal would really take weeks to run if you ran like thousands of bootstraps, or at least days, with a really powerful compute core. FastTree2, you could run really quite fast, really come up with very large trees in a matter of minutes. And I talk to my friends in pharma most days, and I see trees of like kinases and stuff, and really, so I think about how trees are relevant to lots of us. All right, awesome. So, I want to start with a baseline definition of population genomics. This comes from NHGRI. It's a bit dated, but I think it still really fits. And that's really applying genomics and other technologies to understand how genes contribute. Here I have a NovaSeq, and now what we're starting to see, and I'm personally very excited about this, is integration of imaging into our genomic studies, particularly in the context of neurodegenerative disease, which so many of us care about, and we should, because it really affects the population a lot, as well as, not to mention, individual patients. And I think we're sort of slowly falling into the idea of precision medicine. If we start thinking about ALK-positive lung cancers, that's really, I think, a place where we've made a lot of headway, and we will make, and I'll show you some public data around that a little bit later. But my sincere hope is over the next year and 2 years, we can move that into a lot of different disease spaces. And that's really sort of one of the big reasons, I get up in the morning. So hopefully you'll kind of come with me on this journey. So I think that, we've been talking about the right patient at the right time, but again, we've had these algorithms for a while. Why has progress been so slow? And I think that really we've thought about this as sort of a dartboard approach, and really not laid the fundamental groundwork to understand genomes. And I think this kind of illustrates what I mean. I think we need to really start thinking about haploblocks and how haploblocks exist in reference populations versus haploblocks in treatment populations. And that allows us to reduce the search space across the human genome. I mean, a matrix of 6 billion times 6 billion is still really at the edge of our compute power. Whereas if we take sort of largish haploblocks, a matrix of 2300 by 2300 is still somewhat computable. Obviously, for those of you who are theorists, there's a bunch of assumptions there, but I think you get the idea. So let me dive into a glossary, some other stuff for people that are just getting into this, and then we'll get back into like a hands-on demo and then get into the theory at the end. So what is a cohort? Well, you can make a cohort on a bunch of things. Effectively, it is a bunch of individuals, you could call them subjects, with particular phenotypes, with particular genotypes, and now more often particular genotypes and phenotypes. So to go back to the ALK-positive example, folks with, unfortunately, small cell lung cancer and ALK-positive mutations. So that would be an example of an integrated cohort. And my fundamental hope is we'll start seeing and understanding more and more of those cohorts. And even if there's a cohort you can't use, knowing that that cohort is different from the cohort you're interested in, I think is fundamentally important. And then something we think about all the time are genotype and allele frequencies. So, and really understanding linkage disequilibrium in populations is so important, and it's fundamental to this. But fortunately for us, there are lots of really simple to use tools to understand linkage disequilibrium now, and to really put population stratification based on LD into our association models. And that, I think, is important. Also, I really want to underline the idea of haplotypes and phasing. Thinking about clustering haplotypes We also need to really start thinking seriously about admixture and what population haplotypes, looking at mixed populations, as well as data-based relationships between things. And another reason haplotypes are important are because of genetic draft. Now, everybody in this webinar, I'm sure, knows what genetic drift is, fundamental to things, as well as selection. But something we often don't think about is draft, right? So if we have a beneficial allele, we can have a negative, a deleterious allele that actually hides under it, and vice versa. And this is, I think, really important to think about, but we can really only see this if we think about the genome in terms of haplotypes. And so that's really throwing that out there for the theorists for later on. Finally, what is really critical is biological interpretation. A lot of people like to ask me, they say, which algorithm should I use for X in bioinformatics? Which algorithm should I use for Y? And really a mentor of mine in bioinformatics, a guy named Louis Geer, told me once, he said, Ben, you should think about bioinformatics algorithms more as intermediate reagents. Why do you use fish serum to block a gel, right? And that had really a lot of wisdom to it, which is, you can use algorithms, but, a lot of times they're really only as good as what they were tested on. So you want to use a number of algorithms, see what's coincident, and then also be able to back that up with other biological studies. And nowhere is that so important as with machine learning. And we'll talk about that next month. So, but really at the fundamental level of all of this is cohort selection, right? And a lot of that, for those of you who are more advanced, comes back to covariance, which we'll talk about in a little bit. But what we need to be able to do is think about cohort selection. And usually I have historically done most of this on the command line. I'm sure most of you have. If you happen to be a DNA Nexus user or interested in DNA Nexus, we have this very shiny, fancy interface that's available to our Apollo customers, but soon will be available to anyone doing research on UK Biobank. And basically, you can select by sex, by age, by diagnoses, and it's not in this slide, but you can select by medicines and things people have taken, as well as a whole host of other things, including cod liver oil, which I, as an American, find fascinating, that people take cod liver oil, although that may be, my friend Mike tells me that may be an example of selection bias. But anyway, we need to fundamentally select cohorts. And one thing that I think is important, in terms of reducing this activation energy for population genomics, are easy ways to select cohorts. And a lot of our clinician and biologist friends, I actually am a biologist by training, a lot of our clinician and biologist friends are really sort of used to GUI interfaces as opposed to command line interfaces. And tools like this, I think, make that a lot easier to say. And from here, you can really jump off into anything you need to do in bioinformatics. And that's really important, especially if you have sort of GWAS and FWAS tools ready to go. And so if you're not a DNA Nexus user, I would urge you to think about how to set up cohort selection in ways where you can allow your biologist and clinician collaborators to ask the questions they want to ask. So I would say this is really for sort of the more folks newer to population genomics. Really some fundamental sort of tools and strategies. One is GWAS, a group out of Harvard, I believe. The Neil Lab really sort of did some large GWAS studies on a lot of UK Biobank. These are really clever. You can check out what they've done, how they've done it at their GitHub repo, which is really kudos to them for really making this approachable. They use Sage. I'm going to talk about that tool in a little bit. So that's really awesome. And then FWAS, so Marilyn Ritchie, Sarah Pendergrass, and others came up with an approach called FEWAS to look at clinical laboratory measures. So, of course, we think about cancer. Of course, we think about diabetes. Of course, we think about rare diseases. We think about sort of common rare diseases like pancreatitis. But how often do we think about bilirubin in non-liver diseases, right? But that could be really, really critical. So I think FEWAS is a really exciting technique. And my last content slide will talk about thinking about GWAS and then FEWAS and then GWAS, doing these things in iterative cycles. So that's more for you theorists sort of towards the end. But then going back to the newer practitioners, one thing that is absolutely critical is variant annotation. And this is another place the barrier to population genomics has really been decreased. There are lots and lots of tools and databases for variant interpretation. What I use personally most often is OpenCravat. You can check that out at opencravat.org. Obviously, SNP, SIFT, ANIVAR, and then some of these individual databases and tools like ClinVar and Nomad and VEP are clearly very popular, and that's awesome. But I think it's really important to get a biological understanding of what you're looking at so you can tell really what is the noise that's coming out of these other algorithms. Great. So I wanted to talk a little bit about SAGE. So SAGE is, and shout out to the developers of that for making this really quite easy to use. And so with SAGE, you can do GWAS, like I said. That's been done by the Neal Lab, and it's relatively straightforward to look at. We have these results at DNA Nexus, in case you're a DNA Nexus user. I think this population is split about 60-40 from DNA Nexus users and non-DNA Nexus users. So just trying to walk that line. But one thing I'm really excited about that we're providing to our folks, and if you're not a DNA Nexus user, I'd suggest checking this out, is SAGE Gene. So then you can start thinking about regions of the genome and association and really lowering the energy barrier towards thinking about haplotypes and regions. And I will mention explicitly that my clinician friends ask me all the time about the haplotypes in their genes. Now, those of you who are population geneticists know that's not something that actually exists. But I would submit to you that we shouldn't care, because it's so important that clinicians are thinking about haplotypes, and honestly, that they're making the time to think about haplotypes, that we should, for lack of a better word, humor them and really serve them gene level haplotypes. In fact, I just ran a large structural variant hackathon, and one of the most useful projects to come out of that was something we called GeneVar that identifies structural variants from dbVar in genic regions. And really, I think that's helpful to a lot of practitioners, and we should probably think the same way about gene level haplotypes, particularly in association studies. And so one thing to think about a lot is that covariates are so important. And my colleague Deanna Church once said to me, bioinformaticians will almost always take the easy solution over the good solution. And so the word of caution here is, please don't take everything and throw it into a GWAS. Or at least if you do that, please do it iteratively, right? So there's a couple ways to think about this. There's putting in covariates carefully, and I will show you some Jupyter notebooks a little bit later, where you can put in covariates, and that's awesome. And that will help you put in covariates. But also, I think it's really important. Another way to do it is to make lots and lots of different cohorts and then run these in iterative algorithms. So different ways to think about how these things like U affect Y. And I will say that the paper that this comes from is really It's really insightful and really, I think, makes a lot of excellent statistical points, but does not include a GitHub repo where you can replicate their work. And I think that, to me, for those of you who are computational folks, that to me is absolutely critical at this juncture. I mean, in terms of making GitHub repos where you can share things you are putting in academic papers. So please, please do that. And for those of you who are new to this type of thing, please go out and get a GitHub account. GitHub probably won't be the sort of central hub for open source software forever, but currently it is. So please think about joining that ecosystem. So again, I mean, thinking about cohort selection in terms of females who are over the age of 60, this happens to be UK Biobank data, this is synthetic, and who have neoplasms. I've also put cod liver oil, and being able to save those cohorts and iterate over them, I think really, again, reduces this barrier to phylogenetics. And then the nice thing is, if I can also make genotype-based cohorts, and if I make genotype-based cohorts, then I can look at PheWAS, and I can see which phenotype values are associated with various SNPs. And we're going to come back to this later when we talk about haplotype-specific expression. I'd like to point out that Plato, and this is granted, obviously, from the Plato paper, but Plato does a lot of these things. So something to check out. This is provided. It's really easy to use on DNA Nexus. But if you're not a DNA Nexus user, I'd encourage you to check out Plato. And I'm going to show you some Plato results when we get to the demo section. And something to think about later, this is also either in the Plato paper or the subsequent paper written by Pendergrass, Ritchie, and others. But just to think about that, the expression from those particular genes, I think is where we are going in large part now. So that's pretty cool. Also, the more regularized metadata you can get, and obviously metadata is the 800-pound gorilla in the room, but the more regularized metadata you can get into phenotype genotype analysis, the better. And so really, I'm so excited to help out people getting on to looking at the UK Biobank projects, because as you can see, there is just an almost just mind-boggling amount of data that you can look at. And some of that's going to give you noise, and some of those are going to be real covariates, and that's really, of course, what we are trying to pull out of all of this. So this is the DNA Nexus interface. At least 60%, probably more of you have seen it. And as you can see, there's lots of stuff in different folders, so on and so forth. And really, so here we have all of the Neil Labs GWAS results, as well as the 50K synthetic data set. The other thing I can look at are these cohorts. So I can make cohorts real easily and pull them up, and you'll see that in a second. But also I can go back into the data set and I can go ahead and pull up the 50,000 synthetic data set. That just means they're scrambled because I'm showing you in a public webinar, and then we're going to pass it around the recording to this, so on and so forth. And I have these, they take just a second to come up. I have them pre-pulled up, and here you can see, so this is really quite nice. What I can do is I can select, I think that's 60. I can select, as I was telling you before, all, say, males over 60 with diseases of the eye. And that's unfortunate. And then I can see, say, how many of them were taking aspirin. And then calculate the number of folks. And then I can think about what is an appropriate control. Now, yesterday, I was in a session, and somebody asked me about automated controls. And I will tell you that the head of Geo, Tanya Barrett, who's a wonderful person, she asked me for years, can you come up with all the controls in Geo? And obviously, I mean, we had hackathon teams and lots of clever people really think about that problem. And it is a very difficult metadata problem. If you're interested in that problem, email me. But what I will say is really sort of think about putting thoughts into your controls, and you can save that. So I can look at, for example, just to be super simple, I have these 46 individuals. That's not, really big numbers, but males that have eye disease, males over 60 that have eye disease and take aspirin. And if I'm not going to worry about the numbers, what I can do is think about an appropriate control and say, okay, well, an appropriate control might be men who, not pregnancy, who have injury, right? Because we know that probably doesn't have poisoning. Those probably aren't genetically linked diseases, unless, let's not go there. All right, so, and then, yeah, we can recalculate that cohort. Clearly the numbers here are different. That's gonna make the statistics a bit tough, but you can see the general idea here in terms of thinking, and I needed to deselect I, but you see the point. So that's I think, a useful thing. The other thing I can do is go back and look at other GWAS results. So here I have a Plato GWAS, and the reason I want to show you this is to show you really the nice Manhattan plot that comes out of this. Here I'm pulling up Plato GWAS results, and in just a second you will see a Manhattan plot. And the really nice thing is then you can see very easily and really interact with genetics that are genetic things that that are related to your cohort of interest. And I think one thing that's really nice about this kind of approach is you can look at the cohorts a lot. You can do population stratification, put that in there, et cetera, and then really use that and to see what you keep seeing over and over again, and then again annotate variants to look at that. Now, how would I do that variant annotation if I was interested? By the way, I could do the same thing with FEWAS, et cetera. But we are getting along in time. So what I would do is I would go to a Jupyter notebook. And so here you can see that I have a whole bunch of Jupyter notebooks. And so I can look at, I can launch things. I have a Jupyter notebook to make easy covariate files here. Here's some Jupyter notebooks for TCGA analysis. I also have Jupyter notebooks to annotate variants and to do a bunch of other stuff. For those of you who are DNA Nexus users, I also have an API called DX Data, and what I can do is I can slurp data using Spark, which is fancy, into Jupyter notebooks straight from Apollo or from large CSV files, and that's something I really like. So, and if you're not familiar with Jupyter notebooks, please check out the recording from last month on using command line interfaces for genomics. I spent a lot of time talking about Jupyter notebooks then. And if you have specific questions, please put them in the chat or email them to bioinformaticsathome@dnanexus.com, and we can go over them. So, and really, one thing I'm personally excited about here is making a library of Jupyter notebooks such as this available to our research community. And if you're a DNA Nexus customer or part of the DNA Nexus research community, at this point, folks that are using UK Biobank genomic and exome data, please let us know. Email either support or bioinformatics at home, or tweet at me. Feel free to do it publicly about Jupyter notebooks that you'd really like to see and you think are useful for analysis. And I'd love to be able to work with you to make those. All right. So yeah, and this segues into fine mapping well. So GWAS are often difficult to interpret. So for those of you who are younger and had the great fortune not to be around during the advent of SNP chips. Just joking. But so remember that marker SNPs in the chips are not necessarily phenotype associated at all. Remember, they were originally designed to be SNPs that behave reasonably well in populations, which means two things. One, a lot of those SNPs really just tell you about population stratification, even if they're significant in your GWAS. And two, there may be SNPs that are relevant to disease that are very tightly linked to those population stratification SNPs. I worked on a project called Ad Hoc, like the bird, working on teasing these out. But you can also really think about fine mapping for your GWAS. And really, I think this is the next place where we want to lower that activation barrier and really get folks into fine mapping. And Peter Nguyen from DNA Nexus has been working really hard to get this FGWAS fine mapping algorithm working on DNA Nexus. It works great. And so here you can look at really reduce the amount of noise in your GWAS. And I think that's really critical. And so thank you to him. And I'm going to go into the details of this really specifically next month during the Bioinformatics Home Machine Learning webinar. So, but now I'm really going to sort of go off the rails a bit and talk really about advanced theory. So if you're a beginning practitioner, really, I encourage you to log in and check some things out, and really sort of try to maybe hold on here and think about what might be in the future of population genomics writ larger. So this is a project I was involved in an Elixir hackathon, and thanks to Elixir for this, working with a team of really awesome people from Odyssey, NVIDIA, Pitt, a bunch of other places, and of course DNA Nexus. And really what we were thinking about a lot is, if you had a patient or a research subject, how can you go in and say, I think this person may be predisposed to metastasis. How can I check really quickly if they may have some signs of metastasis and refer them for further follow-up? And really, one thing we thought a lot about was that there are definitely population cohorts out there that can be shared by individual researchers. And so how would we align with those population cohorts? So we thought a lot about how we'd onboard that metadata, and we do that through by taking ICD-10 into OMOP and then really comparing variants that you might find in your GWAS with other people's GWASs, as well as a bunch of public databases. So this is something that I think is really interesting. But then what you can do, what's relatively easy to do, and I can pass you the code for this if you want it, is to think about how to flag your VCF. And you can make columns for whether other people's variant sets tag your VCF. And so you can think about maybe a patient reporting system, but also a sub-cohort characterization system. So the idea is to have a whole bunch of those worldwide, and you don't typically have to share patient information unless you want to use graph genomes to boost the signal. And that's actually something that you can do. So for example, and granted this is serial indels, but Eric Garrison and I did an analysis where we used the K562 cell line, and we were able to resolve serial indels in the FHIT gene in K562 as a toxic path through a graph. And the more important thing is you can do that in a mixed sample. So that's pretty exciting. But why should you care? Why should you care about having this constellation of people sharing their data and so on and so forth? Well, the reason I would submit to you is, so I did the same webinar about a week ago, mostly for a sort of West Coast US audience. It was at a different time. And the night before I saw this paper, so thinking about a GWAS that had these 92 novel loci, and what if I'm interested in this type of disease, I probably want to be able to look at those novel loci and compare my data to those loci in a relatively rapid manner. And so that's, I think, one thing that we're working on now. And then literally 1 week ago, this paper came out with 7 new variants for Alzheimer's disease. And what I want to do is be able to annotate my variants looking for any of those new loci that are in late onset Alzheimer's disease. And so that's, I think, why we want to be able to really compare our population genomics results to other people's population genomics results in real time. And really, I don't want to de-emphasize the importance of ClinVar and other sort of large databases, but the funnels for data to get into those resources are relatively tight and relatively slow. Whereas if we can share expressed variant sets with each other, we can start going really fast. So I want to bring some of these advanced concepts that I've been alluding to together, really thinking about the characteristics of driver haplotypes and haploblocks and risk haplotypes and haploblocks. And that's really, I think, what we're typically driving for on GWAS when we're doing GWAS and other similar analyses. So remember this. So this comes from really sort of the FIWAS paper. And what we see is that they really took an early look at expression of haplotypes and used that to prioritize what was most important. And really, now it's relatively easy to look at linkage disequilibrium as well as haplotypes. So check out LD-Link, similar tools, as well as WhatsApp. And then Phaser from the Lapidin lab is something that I've been really excited about for a while, because not only will it do phasing, but it will also tell you about expression in those phased genomes. So 3 tools to check out. And by the way, kudos to my friend MedHat at Baylor College of Medicine for making WhatsApp into a DNA Nexus applet. So if you're a DNA Nexus user, ping me. I'll send you over the applet for WhatsApp. Written by, oh man, I'm blanking on the name of the group that wrote WhatsApp. I apologize. And if they're on the call, my fault. So anyway, yeah. And so I think it's important to think about expressed haplotypes, but what you might say is, well, I have a large population that I have exome or even whole genome for, but I don't have RNA-seq. So what do? I can't go back and reconsent those people for RNA-seq. And you're right, you can't. But if you think about it, if you have a pivotal SNP that you think is critical, and that is in a coding region or in a UTR, overlaps with a coding region or UTR, then you can see if expression is different in that in that SNP versus not in that SNP. You say, well, where do I get that RNA-seq data? Well, let's go back to that lung cancer example. Check this out. There are 34,000 RNA-seq data sets on lung cancer in the SRA. So you have lots of data to work with when you ask the question that way. Now, is that the perfect way to ask this question? No. What do I want? I want WGS with RNA-seq for every patient, actually at a bunch of time points. Am I going to get that right now? No. But, you know, I mean, that's the future we want to work for. But before that, we have to convince the powers that be that these approaches are clinically applicable. And by doing these types of analysis, I believe we are going to do that. Oh, by the way, you're probably saying, well, I have RNA-seq. How do I get the variants to match with the other variants? Check this out. So you can, GATK will call variants from RNA-seq. This is something that a lot of my friends are doing now. I'm very excited about it. And actually, if you go to DocStore, thanks to Brian O'Connor for this awesome resource, you can really click on the DNA Nexus button and launch this workflow on DNA Nexus. It's a pretty easy thing to do. So check out DocStore. You should be able to just Google for it. And you can always just grab the Docker container if you want to do something funky with it or anything. So that should work out pretty well. And then, so yeah, I think it's really pretty critical to do this, and folks have really worked on this. And I think this is really sort of in its nascent phase, but all of the constituent tools are much easier to use than they were even a couple of years ago, and that's something that's exciting. And if you're a DNA Nexus user, they're really quite easy to use. So then I want to get to the sort of a last point on that. So I call this the DJ project because really sort of it was founded by my friend Doman. And this is not a picture of Doman. This is actually a picture of my colleague Momin. It's totally coincident that their names rhyme. But anyway, so I the concept here is, and this is really a new research project, is to do GWAS, then PWAS, then GWAS, and then compare the GWAS to see if the covariate sets are sufficient. And I think that's pretty exciting because then if overlap is low, then in the PWAS, you should be able to see what you're missing in terms of covariates and work them in. And what is likely is that some of these results are going to be pretty confusing. You may have pleiotropy, which is sort of exciting for drug repurposing, or you may have crazy methylation going on in the genome. But the most critical thing for all of this is data provenance, to know what cohort you made, how you made it, what covariates you're using, all of this. And you don't really, in my personal opinion, I mean, having done it for many years, searching back through your command line data sets with grep is not a great answer to data provenance. And so thinking about having a data provenance system at your fingertips, I think, is really critical for applying these somewhat cutting edge techniques to population genomics. With that, I'd like to thank a bunch of people. It looks like we are right on time. And I would like to take any questions you may have.
Thank you so much, Ben. Again, you can drop your questions in the chat window or the Q&A at the bottom of your Zoom screen. All right, Ben, I've got our first question for you. During cohort selection, how do you think of balancing pre-filtering out known linkage and over-filtering and missing a novel finding?
Well, so, I mean, I think there's, again, as I said in the webinar, I think there's a relatively simple answer to that, which is to be able to make your cohorts fast and easily, right? If I'm locked into the cohort I made because I put a ton of work into it, and I'm doing a GWAS and it's like this person, you know, my like collaborating lab or my competing lab did a GWAS and they use this set of covariates, and I have to use exactly that set of covariates, so I'm kind of locked into a cohort and locked into my covariate set, then I'm probably going to miss things. I mean, I'm definitionally looking under the lamppost, right? But if I can make covariates rapidly and it's easy to feed them into my analysis using Plink or whatever. By the way, Plink is easily accessible on DNA Nexus. So I can feed things in, and sorry, I was looking at the chat for a minute. If I can easily make cohorts and covariate sets and feed them in, then I am no longer locked into looking under the lamppost, and therefore I am less likely to miss things.
Great, thank you, Ben. Oh,
yeah.
Yeah, I've got the next question in the chat here for you, asking for another brief overview of the DJ project, as you mentioned, the GWAS, DeFIWAS, and GWAS.
So I'm so glad you asked about it. And so the idea there is that, so say you have a set of, you know, I mean, people over 50 with neoplasms, right? So you do a GWAS on those folks, right? And you put in some covariates, and then you say, okay, well, I put in my covariates, I got some significant SNPs, actually a bunch of significant SNPs, but I think I'm missing something. So what I want to do is take all of those significant SNPs and run FEWAS on them, right? And so I run FEWAS on all of them. And what I want to see is, okay, now I have a bunch of new covariates. And then what I can do is put in those covariates. I can put in those covariates and run new GWAS based on uh, selecting for those cohorts. And that is something that I'm pretty excited about, because then really you should be able to pull out more salient SNPs, both putting them all in, but also bootstrapping sort of a leave one out type of approach. Say I came up with 2 or 3 more. Yeah, so that's what we're thinking, is that the phenotypes associated with the SNPs are new covariates. And if you want to talk about this offline, please feel free to tweet at me or send me an email. I'd be happy to talk about it more. And like I said, that's an ongoing project, so we'd be very interested in working with collaborators if that's something you're interested in.
Fantastic. Thank you, Ben. Next question. Why not do a GWAS for all variants in dataset, or like a large scale dataset like UK Biobank? Are there any pitfalls with that?
Well, so I love this question, and honestly, the Neil Lab has really done a lot of work to address that question. But I think there is a word of caution here, particularly for young data scientists, to say, well, I can throw everything in a bag and get everything back out. And one of the reasons for that is because the space of phenotypes and like subphenotypes and environmental exposures is massive, right? So to try to say I can calculate over everything at the same time really doesn't make statistical sense. I think at circa late 2020, and this may be no longer true in a year or two, but right now, I think what makes a lot of sense is to come up with the smallest buckets that we can that are still statistically relevant, and compare between those instead of trying to swallow the entire ocean. I think really what we've seen with those types of projects, with some caveats like the Neil Lab work, for example, is that really you end up with a lot of noise.
Thank you, Ben. All right, I think our last question here is where can I find more information about the DNA Nexus Core browser that you're showing off today?
Right. So yeah, if you just check out the DNA Nexus website, that's there. And then again, we have a research program right now for UK Biobank, or soon for UK Biobank, where all UK Biobank folks will, all approved UK Biobank applications will be able to work with Apollo. So either contact DNA Nexus about grabbing Apollo. Some of you actually are probably DNA Nexus users. You use our core platform. You don't use Apollo. Talk to whoever your sales folks are about getting an Apollo demo. And you can have your cake and eat it too. So you can use it for UK Biobank, and then when you're ready, say you have a 30,000 WGS or 6,000 WGS that you want to 100 WGS that you want to put into Apollo, or even just phenotype imaging, whatever it is, let us know, and we'll work to get you set up with Apollo. But that's that it should be relatively straightforward. And again, if there are Jupyter notebooks, that kind of thing you'd like to see, let us know.